Purpose of this video tutorial will be to introduce you to some of the basic aspects of the immune system and how the innate and adaptive immune system work together to eliminate pathogens. So we are surrounded by microbes all over in our environment. And most of these microbes, including bacteria, viruses, fungi, and parasites, are relatively benign. They're commensal organisms that interact with our bodies in non-harmful ways, or they're environmental microbes that really don't want to have anything to do with us. But a very small subset of these microbes are pathogens, and these are microbes that cause disease. And for this reason, we have evolved an immune system to be able to combat these pathogens and prevent disease. The key function of the immune system is first to distinguish between self and non-self, and this helps increase protection from things like infectious pathogens, but also other things like tumors. But the immune system also wants to make sure that it is only inducing an immune response to foreign things, and so this ability to distinguish between self and non-self decreases the chance of autoimmune reactions developing. The immune system also has to determine the difference between things that are dangerous versus things that are benign. And so we'd like to make an immune response to pathogens, but not necessarily an immune response to harmless organisms like commensals or other harmless antigens like food. So as we get into this tutorial, there's some key definitions that I will be using that are listed here. Take a moment to view these definitions so that you understand what the meaning of these terms are as we use them throughout this tutorial. So white blood cells, or leukocytes, all originate in the bone marrow, and they are all derived from hematopoietic stem cells. Hematopoietic stem cells give rise to two important lineages of leukocytes. The first are the common lymphoid progenitors and the common myeloid progenitors. The common lymphoid progenitors give rise to lymphocytes, including T cells and B cells and natural killer cells. And they also give rise to an interesting cell type that we'll learn more about later called the dendritic cell. Common myeloid progenitor cells, in turn, give rise to myeloid cells, and that includes things like neutrophils, eosinophils, basophils, mast cells, and monocytes. And they can also give rise to some dendritic cells, and so this is a complicated cell type. And again, we'll learn more about dendritic cells later. Now, of the myeloid cells, neutrophils, eosinophils, basophils, and mast cells are classified as granulocytes, and more specifically, neutrophils, eosinophils, and basophils are often referred to as polymorphonuclear cells because of the odd and segmented formation of their nuclei. And lastly, monocytes are found only in circulation, but as they enter into tissues, they can differentiate into another cell type that's very important that we will talk about throughout this tutorial called the macrophage. Now, these cells participate in the immune response, and broadly, the immune response can be divided into two different phases. The first is the innate immune response, and the second is the adaptive immune response. And I'd like to take a moment just to compare and contrast these immune responses. The innate immune response provides barrier protection. So in some ways, we could consider our skin and our mucosa and our epithelial cells as part of our innate immune response because they provide a barrier between us and the organisms in the outside world. But some key differences also occur that help us understand the innate and adaptive immune response. Innate immune responses happen immediately, within minutes to hours after exposure to a pathogen. And this can involve cell types such as myeloid cells and natural killer cells. Whereas adaptive immune responses take several days to get going and typically involve B cells and T cells. Within the innate immune response, there are a number of important secreted molecules, including complement, defensins, and other molecules that mediate immediate responses to pathogens and eliminate them and lead to their destruction. The adaptive immune response also has secreted molecules, and these secreted molecules include antibodies and cytokines, which we're going to learn a lot about. Now, the innate and adaptive immune responses also differ in their antigen receptors. Innate immune cells have antigen receptors that are encoded in their DNA, meaning that all innate cells typically have the same range or same receptors. And these receptors have broad specificity for commonly expressed molecular motifs within pathogens. The adaptive immune response, on the other hand, generates immune receptors through a process of DNA recombination, meaning that every single T cell and every single B cell has its own unique receptor, and these receptors are very finely specific for pathogens. Whereas the innate immune response can make a broad response, the adaptive immune response is very finely tuned to the pathogen that you're infected with. And one really important aspect to this is whereas the innate immune response does not induce immunological memory, the adaptive immune response does. What that means is if I get infected with a pathogen, my innate immune response will form a response. And if I get infected with that same pathogen 6 months later, my innate immune response will form the same response. But my adaptive immune response has memory. Every time it's exposed to that pathogen, its response gets better. Okay, now what are the cells of the innate immune response? Well, I mentioned many of these myeloid cells, and that includes things like neutrophils and monocytes and macrophages. The difference between neutrophils and monocytes to macrophages is that macrophages don't circulate. They're really only found in tissues. And these cells are important in many types of immune responses, including against many bacteria and fungi and parasites. Natural killer cells are fairly small subset of leukocytes and are important in the antiviral response. And then the eosinophils and the basophils and the mast cells are important in responses to certain types of pathogens, including helminths or worms that you'll learn about later in your education. And they're also important mediators of the allergic response. Cells of the adaptive immune response are essentially T cells and B cells. And T cells in particular interact with dendritic cells. And dendritic cells are important for activating T cells. They capture antigens and present them to T cells to activate them. And again, these responses are highly specific to all types of pathogens. So how does an immune response get started? Well, here's let's call this a skin tissue in which macrophages and dendritic cells are resident. And if there's some cut or some way in which pathogens are introduced into that skin, and here I've shown just some bacteria that have gotten into the skin via this cut, they can interact directly with macrophages. Macrophages have receptors that can recognize the presence of these bacteria, and they start to produce a class of molecule called cytokines that produce inflammation. At the same time, dendritic cells can also be activated by exposure to these bacteria, and they will get out of that tissue, migrate to a draining lymph node, and activate T cells. And lastly, there's a class of molecules called complement that mediate inflammation and pathogen destruction, and these molecules are also activated by exposure to these bacteria. Okay, so let's talk about complement for just a moment. Complement is a rapid response system to foreign pathogens. It's a group of more than 30 proteins that are constitutively in the plasma. These are proteases, and they circulate in an inactive form, but when they are exposed to a pathogen, they become active. This leads to a proteolytic cascade, leading to the generation of many different fragments that have diverse biological effects. And among these effects are destruction of the pathogen and induction of inflammation. Okay, so complement fragments are playing an important role in driving inflammation within this environment. Now, let's talk a little bit now about the inflammatory cytokines that are produced by macrophages. So what are cytokines? So cytokines and a subclass of cytokines called chemokines are small secreted proteins. They control growth and function and activation of immune cells that express the appropriate cytokine receptor. The cytokines can be constitutively expressed or inducibly expressed. They mediate communication, and a subset of cytokines are chemokines, and these are cells that mediate chemotaxis, or movement of cells from an area of low concentration of chemokine to an area of high concentration of chemokine. We're not going to talk about the identity right now of individual chemokines or cytokines. I only want you to know that there are some cytokines that are inflammatory, and there are some complement fragments that are inflammatory. And when these complement fragments and inflammatory cytokines get produced after exposure to a pathogen, they will act on the local capillaries to activate them and induce their vasodilation. And the effect of this vasodilation is the migration of cells in circulation across these capillary walls into the inflamed tissue. So here's an example of a neutrophil that's migrating in the capillary, and when it comes across this activated endothelium that is vasodilated, it can squeeze in between these cells into the infected tissue. Now, once these neutrophils are in the infected tissue, they can migrate along a chemokine gradient. Here's some macrophages that are being exposed to some pathogens, and they're making chemokines, and so they migrate to the source of those chemokines, and that is what chemokines do. They force these neutrophils to migrate to where the pathogens are. And this is essentially the process that leads to an inflammatory response. So when you get a cut and that cut starts to inflame and swell, that's because you've got chemokines that have led to neutrophils coming into that tissue and causing an inflammatory response. Okay, so what are some of the effector functions of these innate immune cells? Well, they are good phagocytes. And what a phagocyte is a cell that eats stuff in its environment. And so here's a phagocyte that has eaten some bacteria and drawn it into this structure called a phagolysosome, where it can employ a variety of mechanisms to destroy these bacteria within the phagolysosome. Macrophages and neutrophils are classic phagocytes. Some of these cells have cytotoxic granules, and when they get activated by exposure to pathogens, they start spitting these granules out. And these granules are highly inflammatory and have a number of effector molecules that are toxic for pathogens. Many times these cells die, but neutrophils have a specialized function that when they die, they release their DNA, and this creates a sticky matrix that traps bacteria and allows for other cells of the immune response to swoop in and kill them. And lastly, some cells can be killed directly by cells of the innate immune system. So for example, natural killer cells are really good at finding and detecting virally infected cells and eliminating them to prevent spread of the virus. Okay. All right, so we've talked a little bit about activation of macrophages and activation of dendritic cells in complement. Let's turn our attention to dendritic cells and ask what it is that they're doing. So dendritic cells, when they're exposed to pathogens, are also phagocytes. And so they've been taking up these pathogens into phagolysosomes. And when they become activated by exposure to pathogens, they migrate to a lymphatic vessel. And within that lymphatic vessel, they're going to migrate towards a draining lymph node. And within the draining lymph node, they can activate T cells. And so here's a dendritic cell that has migrated to a draining lymph node. It's going to be able to present antigens to different types of T cells. That's going to lead to their activation into either helper T cell subsets or killer T cell subsets, also known as cytotoxic T lymphocytes. Helper T cell subsets are really important for secreting inflammatory cytokines, for activating a bunch of innate cells. They're important for having antibacterial functions, antiviral functions, antifungal functions, antiparasitic functions. Basically, helper T cells are always activated in response to virtually every pathogen. CD8 T cells are killers, and so they directly kill virally infected cells and are particularly important for the antiviral response. B cells, on the other hand, get activated by soluble antigens or free-floating antigens that migrate via the lymphatic vessel into the B cell area of a lymph node. And when B cells get activated by antigens, they become plasma cells that secrete antibodies. And these antibodies, which we'll learn more about in subsequent tutorials, are very important molecules that can neutralize pathogens and prevent them from infecting. So an antibody can bind to the surface of a virus, for example, and prevent that virus from infecting its target cell. Okay, that is the end of this intro lecture. This has been a brief introduction to some of the functions of the innate and adaptive immune response, and an introduction to how they eliminate pathogens.